respiration, we have seen that there are two types of respiration broadly classified as external and internal respiration. In the internal respiration, where it takes place inside the cells, so the respiration is also called as cellular respiration. So cellular respiration is again divided into aerobic and anaerobic. Under aerobic, we are studying three steps: glycolysis, Krebs cycle, and oxidative phosphorylation. Now in the cell, suppose this is a nucleus and this is the mitochondria. So the glycolysis takes place in the cell, in the cytoplasm, where the glucose is converted into pyruvate. This pyruvate will enter into the mitochondria, where the second and the third steps takes place. So the second step, Krebs cycle, takes place in the mitochondria of the matrix. The third step, that is the oxidative phosphorylation, takes place in the membranes of the cristae, which where the Racker's particles are located, or the elementary particles are located. So in glycolysis, which is also called as EMP pathway, glucose is converted into pyruvic acid. Pyruvic acid enters into the mitochondria. In the matrix of the mitochondria, the Krebs cycle takes place. Krebs cycle is also called as citric acid cycle or TCA cycle, tricarboxylic acid cycle. Now then, the NADH2 and FADH2, which is formed in the Krebs cycle and glycolysis, they enter into the Racker's particles of the mitochondria. Now in the crystal membranes or the Racker's particles, there are electron carriers. So this is the third step of the aerobic respiration. So oxidative phosphorylation, and here phosphorylation means we already studied ATP is formed by the addition of inorganic phosphate to ADP. So the phosphorylation takes place due to oxidation here. Now in photosynthesis, there is photophosphorylation. The ATP formation takes place due to the influence of light, and oxidative phosphorylation takes place due to oxidation. So in the crystal membranes or the Racker's particles, there are cytochromes. So these cytochromes are electron carriers. So there is cytochrome B, there is cytochrome C, there is cytochrome A, there is cytochrome A3. So B, C, A, A3, these are the electron carriers present in the mitochondrial membranes, that is the crystal membranes. So here what happens, during glycolysis there are 2 NADH2. During Krebs cycle there are 8 NADH2. So total 10 NADH2. And during Krebs cycle there are 2 FADH2 produced. There are 2 GTP. Like that, the NADH2 and FADH2, they undergo oxidation. So NADH2 will release the electrons to form NAD. So totally there are 10 NADH2. Out of that, each NADH2 undergoes oxidation, it releases the electrons. Actually, this H2 will split into 2 electrons plus 2 hydrogen ions. So these electrons are accepted by cytochrome B. Now these electrons actually contain energy, and these electrons, when they are accepted by cytochrome B, at this step they release certain amount of energy. The energy released is shown in this way. So that energy is taken by ADP to form ATP. So one ATP is formed at the first step. When NADH2 is oxidized to form NAD, so the electrons are released. The electrons are accepted by the first electron carrier, cytochrome B. So this is a sequence of electron carriers. So oxidative phosphorylation involves a sequential series of electron carriers, cytochrome B, cytochrome C, cytochrome A and cytochrome A3. And then cytochrome B will receive the electrons. Before that, the electrons release a certain amount of energy that is accepted by ADP to form ATP. So then the electrons, they pass to the next cytochrome. So these are the electrons. So similarly, they pass on to the another next cytochrome, and they pass through the series of electron carriers. So when they pass through a series of electron carriers, now here the electron will release another little amount of energy, and that is taken by ADP to form ATP. So ATP is formed. Similarly, when the electrons pass from B to C, C to A and A to A3, here another little amount of energy is released, and that is again taken by ADP to form ATP. This is how the ATP is formed. So one NADH2, by oxidation, the electrons pass through the electron carriers, through the sequential series of electron carriers. So during the movement, they release energy at different steps, and this energy is accepted by ADP to form ATP. So one molecule of NADH2 will produce one ATP. So that means one NADH2 will produce 3 ATP. Similarly, when one FADH2 produces undergoes oxidation, it produces 2 ATP. So totally here NADH2, there are 10 NADH2, there are 2 FADH2. So totally there is 30 ATP and there is 4 ATP produced here. So this is the ATP produced by NADH2 and FADH2. And the GTP which are produced, so during Krebs cycle, so 2 GTP are formed, which are nothing but they are ATP, equivalent to ATP. So totally we get 36 ATP from one glucose molecule. So when one glucose undergoes oxidation, it produces 36 ATP. But however, there are 2 ATP during glycolysis. When we mentioned about the end products of glycolysis, there are net gain 2 ATP. Actually 4 ATP are produced, 2 ATP are utilized. So we get 2 ATP net gain. So that 2 ATP are used in the transportation of the pyruvate. They are used in the transportation of the pyruvic acid. So that pyruvate is transported to the mitochondria using that ATP. So that means at the end of the aerobic respiration, after the 3 steps, those 2 ATP will not be available. That is why whatever ATP we get during a Krebs cycle and during the oxidative phosphorylation, so they are to be considered the 2 ATP which are produced during the glycolysis. They are used in the transportation of the pyruvate molecules to the mitochondria. So here totally 10 NADH2 are produced, 2 NADH2 during glycolysis, 8 NADH2 during Krebs cycle. So those 10 NADH2 will produce 30 ATP. Each NADH2 produces 3 ATP after oxidation. So during this oxidation, one more point is, there is also a compound which is involved in this step, which the mentioning is not necessary. Flavin mononucleotide is a compound which involves at the oxidation step of NADH2, where it helps in the transfer of the electrons to the cytochrome B, and the protons are separated. So there FMN is involved in that step, in the oxidation step. So the 10 NADH2 produces 30 ATP, 2 FADH2 produces 4 ATP, and 2 ATP are produced during Krebs cycle in the form of GTP. So GTP is equivalent to ATP, and thus totally we get 36 ATP from one glucose after the oxidation of the 3rd step. So here the electrons after the cytochrome A3, so what is the fate of the electrons? So where they go? And actually these 2 electrons which are there, they again combine with the hydrogen ions to form H2. So H2 is formed. So this H2 which is formed is not hydrogen gas. It is combined with the half oxygen, means a single O, nascent oxygen. So it will combine with this single molecular oxygen, and when it combines with O, it produces H2O. So H2 plus O will produce H2O. That is how water is produced in the oxidative phosphorylation. So if you go through the equation, the C6H12O6 combines with the 6O2. This is the 6O2 which is used here, and it gives rise to 6CO2. That 6CO2 is released during the Krebs cycle. 6H2O is produced, and energy is produced. This is the energy. So here, to recall the equation here, 6 oxygen is used, 6 CO2 is released, 6 H2O is released, and ATP energy is released in the process. This energy is nothing but the ATP molecule. So glucose after oxidation, oxygen does not combine directly with glucose. Glucose is converted into pyruvate during glycolysis. Pyruvate enters into the mitochondria, where it undergoes reactions, and these reactions produce NADH2, FADH2 and GTP molecules during the Krebs cycle. And then in the 3rd step, the NADH2 and FADH2, they undergo oxidation, and after oxidation they produce ATP. So the electrons after formation of the energy, so here this step is sometimes called as the terminal oxidation. Means at the end, the oxidation results in the formation of water. So cytochrome A3 will donate the electrons to the protons to form H2 and combine with the O, and then it forms water. So this is the water which is formed during the respiration reaction, and this is the energy which is produced during the respiratory reaction. This CO2 is released during the Krebs cycle. Oxygen is used here. So only in the third step actually oxygen is used, not in the first step. In the first step, glycolysis takes place completely without the use of oxygen in the cytoplasm. The second step, Krebs cycle, takes place in the matrix of mitochondria, and it takes place only if there is oxygen in the cell. But however, oxygen is not directly used in the Krebs cycle. So then in the third step, oxidative phosphorylation, oxygen is used to produce the molecule of water. That is, we call this as the terminal oxidation. And this step is also called as the electron transport system, ETS, because the electrons are transported in a system. The electrons are transported in a peculiar or a specific sequence in a particular series from cytochrome B, C, A3. So this terminal oxidation takes place in the Racker's particles or in the elementary particles of the mitochondria. So that is about the aerobic respiration. So these are the three steps of aerobic respiration. All the three steps carry 5 marks. Glycolysis, schematic representation, then Krebs cycle, schematic representation, that is also 5 marks, and this oxidative phosphorylation is also a 5 marks question. It is also called as the electron transport system, or sometimes it is the terminal oxidation, where the ATP is produced. Now we shall see the anaerobic respiration. So anaerobic respiration, it takes place without the use of oxygen. Now anaerobic respiration or fermentation. So fermentation, anaerobic respiration without use of oxygen in some microbes like yeasts and bacteria, in some bacteria and also in some the yeasts. Then here glycolysis is the first step also. So glycolysis is common to aerobic respiration as well as anaerobic respiration. So both. Now this anaerobic respiration is of two types based on what is the end product of the anaerobic respiration. So sometimes during anaerobic respiration in some microbes the end product is alcohol, ethanol actually. So it is alcohol fermentation. So this is one type of fermentation, and there is one more type of fermentation called as lactic acid fermentation, lactic acid anaerobic respiration or lactic acid fermentation. So alcohol fermentation where the end product is ethyl alcohol, lactic acid fermentation where the end product is lactic acid. Now in alcohol fermentation, what happens? Glucose is converted into pyruvate. Glycolysis takes place here also. The same, what we studied in the aerobic respiration, the same sequence of reactions takes place. glucose is converted into glucose 6-phosphate, then glucose 6-phosphate into fructose 6-phosphate. Fructose 6-phosphate is converted into fructose 1,6-biphosphate. Now this fructose 1,6-biphosphate undergoes cleavage. It will split to form triose phosphates, glyceraldehyde 3-phosphate, which may convert into its isomer dihydroxy acetone phosphate. Then the glyceraldehyde 3-phosphate is oxidized to form 1,3-biphosphoglyceric acid, where NAD is converted into NADH2, and there is addition of free phosphate. So then this 1,3-biphosphoglyceric acid is converted into 3-PGA by the release of 1-phosphate. So this 3-PGA is converted into 2-PGA by the shifting of the phosphate from the third carbon to the second carbon. So 2-PGA undergoes hydration, removal of water, to form phosphoenol pyruvate. Then phosphoenol pyruvate releases the phosphate, where ATP is formed, so that ATP means PEP, phosphoenol pyruvic acid, is converted into pyruvate. So that is how the pyruvate is formed during glycolysis. Same way the glycolysis takes place in the alcoholic or in the fermentation. Glucose is converted into pyruvate. Now this pyruvate undergoes decarboxylation. So pyruvate is a 3-carbon compound. So this is a 3-carbon compound. Now this pyruvate undergoes oxidation reduction. That means it is added with the NADH2 to give rise to CO2 to form ethyl alcohol plus CO2 plus NAD. So NADH2 is reduced to is oxidized to NAD. Pyruvate is reduced. The electrons of the NADH2 are accepted by pyruvate, and there is decarboxylation. So the 3-carbon compound forms 2-carbon compound. That is in the case of alcoholic fermentation. So ethyl alcohol is produced as the product in this reaction. So CO2 and NAD. So CO2 is released in this. There is a decarboxylation and reduction. So pyruvate undergoes reduction and decarboxylation. That is in the alcohol. In the lactic acid, what happens? Again, the glucose is converted into pyruvate by the process of glycolysis. So glycolysis, during this, the glucose is converted into pyruvate. So then pyruvate does not undergo decarboxylation. Instead, it undergoes only reduction by NADH2 to form lactic acid plus NAD. So this is also a 3-carbon compound, and this is also a 3-carbon compound. There is no decarboxylation. So the only difference between these two is, here there is decarboxylation, here there is no decarboxylation. That means there is removal of carbon dioxide, there is no removal of carbon dioxide. So when there is only reduction, the end product will be lactic acid. If there is reduction and decarboxylation, the end product is alcohol. So that based on what end product is produced, there are two types of fermentation. So the alcohol fermentation and the lactic acid fermentation. So this alcohol fermentation takes place in some yeast cells, and this lactic acid fermentation, so it takes place in some bacteria like lactobacillus. So the conversion of the milk into curds, it is the example of lactic acid fermentation, where actually lactose is converted into lactic acid in the milk. So thus the milk is converted into curds. So alcohol fermentation is taking place in yeast cells. So yeasts, by the fermentation of glucose, they produce alcohol. Bacteria, by the fermentation of the glucose carbohydrate, they produce lactic acid. That is about the fermentation which takes place in some microbes. Then if we come to the other part of the this respiration, so respiration is also called as amphibolic pathway. Respiration is called as amphibolic pathway. Now what is this amphibolic pathway? We shall see here. So amphi means dual, double. Amphibians we call it as. So here what this amphi is related to. So we know that what actually takes place in anabolic reaction and what takes place in a catabolic reaction. Now anabolic reaction means it is building up complex substances from simple substances. So the formation of the complex from simple is anabolic. Now catabolic means the complex are converted or broken down into simple or simpler substances. That is catabolic reactions. So photosynthesis is a anabolic reaction which we can previously studied. So CO2, water is used, which are simple molecules, simple inorganic molecules. So the CO2 gas, the water molecule is used in the formation of the complex sugar, that is carbohydrate. So this is purely an anabolic reaction. But when we see the respiration, glucose is converted into different compounds. So glucose is converted into carbon dioxide, water and energy. So but this respiration, when we observe the reaction, it appears to be catabolic reaction. The complex compound is converted into simpler compounds. But when we thoroughly go through the steps of the reaction, especially the aerobic reaction, so the respiration also involves anabolic process, anabolic steps. So there are anabolic steps in the respiration. So that means when we observe the Krebs cycle, in the Krebs cycle, the pyruvic acid is converted into acetyl coenzyme A. The acetyl coenzyme A combines with the oxaloacetic acid. Then oxaloacetic acid will form citric acid. Citric acid will form further alpha ketoglutaric acid and so on. So in the initial steps, just when we observe, oxaloacetic is a 4-carbon compound. Acetyl coenzyme A is a 2-carbon compound. Now these two compounds, they form a 6-carbon compound which is more complex than this, that is citric acid. So when you observe this step, it seems to be a anabolic step where simpler compounds have been converted into more complex compounds. So, but overall when we see the respiration reaction, it is catabolic. But when you observe the steps of the Krebs cycle, it indicates that it is anabolic reaction. So respiration involves both catabolic and anabolic. But however, glycolysis is catabolic. Glucose is converted into pyruvate. Glucose is a complex, pyruvate is a 3-carbon compound, so it is purely a catabolic step. Glycolysis is a catabolic. But only thing in the Krebs cycle, we come to know that the pyruvate, which is a 3-carbon compound, will form citric acid by the formation of acetyl coenzyme A. So this acetyl coenzyme A combines with the oxaloacetic acid to form citric acid, which is to be a anabolic reaction. So as respiration involves catabolic and anabolic steps, so it appears to be having anabolic steps and also catabolic steps, hence it is called as a amphibolic pathway. So when it is purely building up a process, it is anabolic. It is breakdown process, catabolic. Building up means simple molecules are converted into complex, build up. So the simpler are built up to form complex. But when the complex is broken down to convert into simple, it is catabolic. But respiration involves both catabolic and anabolic. So whatever the portion of the anabolic and catabolic may be, but however respiration also involves, it shows that it is also having anabolic steps. Hence, as the respiratory process or the aerobic respiration, it shows catabolic reaction and anabolic reaction. Hence, it is considered as a amphibolic pathway. Amphi means dual, double. Amphibians, animals living on land and water, both. When they are purely in water, aquatic animals we call it as. When the animals are purely on land, terrestrial. But frog, amphibians, they live both on land and water. So they are amphibians. Amphi means double or dual. So this respiration exhibits both dual type of reaction, anabolic and catabolic. Hence respiration, cellular respiration is considered as anabolic, the amphibolic pathway, as it indicates or it shows both anabolic and catabolic type of reaction. So it is considered as a amphibolic pathway. This is what is amphibolic pathway. Then if we come to another aspect called as respiratory quotient, or which is also called as RQ value. So the RQ value, when we observe in a respiratory reaction, we come to know what is this RQ value, especially in a aerobic reaction. So when glucose is oxidized in the presence of oxygen, CO2 is produced, water and energy. Now consider how much volume of oxygen is used, how much volume of carbon dioxide is released in a reaction. However, we have mentioned here 6 O2 and 6 CO2. This is 6 molecules. Now, when you consider the volume, they are also almost same. So, when we consider the volume of a reaction, means the volume of oxygen consumed in the reaction, to the volume of CO2 released in the reaction. If you calculate this value, so this ratio, the ratio of the volume of oxygen consumed to the volume of CO2 released in a respiratory reaction is called as RQ value. This is called as RQ value. So what is RQ value? RQ value is the ratio of the volume of oxygen consumed to the volume of carbon dioxide released in a respiratory reaction. So for a carbohydrate, if you take for any carbohydrate, when the respiratory substrate is carbohydrate, the RQ value is always 1. So here this is a carbohydrate. So here the volume of oxygen, the volume of carbon dioxide are equal. So when the volume of oxygen consumed and the volume of CO2 released are equal, so the value will be automatically 1. Now similarly, if you take for proteins and fats, the RQ value is less than 1. So when the RQ value is less than 1, it means that the oxygen consumed will be less, the carbon dioxide will be more. So that means when the oxygen consumed will be less than the carbon dioxide released, so then definitely it will be less than 1. And similarly for organic acids, the RQ value will be more than 1. So this is for organic acid. So for organic acid it will be more than 1. So when it is more than 1 means the oxygen consumed will be more than that of the carbon dioxide released. So that means it will be somewhat 1.2, 1.3, 1.4 like that. The oxygen consumption will be more and the comparative to the carbon dioxide which is released in the reaction. But for proteins and fats, it is always less than 1, where the oxygen consumed will be very less, and whereas the carbon dioxide released will be more, so the value will be automatically less than 1. So for organic acids means there are certain organic acids which act as respiratory substrates. For example, the stearic acid, lauric acid, linoleic acid, and even citric acid, all these organic acids, they act, or they are respiratory substrates. They enter into the respiratory reaction and also they produce energy in the respiratory process. So this is the CO2 actually which is released during the respiration. So the oxygen what we take actually, it enters into the that is the lungs, for example in the external respiration. So that oxygen enters into the cells where it is used in the oxidation process. Food is oxidized and that oxygen, after it is used, the cells produce CO2. That CO2 is carried back to the lungs, and during expiration we release the carbon dioxide during the external respiration. That is how the respiration, cellular respiration which takes place. So we refer this as the chapter in respiration in plants. So this respiration not only takes place in plants, but it is studied under this title to avoid confusion with that of the respiration in the human physiology. where we study the external respiration. So this cellular respiration is again divided into aerobic and anaerobic. So in the aerobic, we studied that there are 3 steps in aerobic respiration: glycolysis, Krebs cycle, and oxidative phosphorylation. So glycolysis, already we have studied glycolysis. During glycolysis, glucose is converted into pyruvate. So this pyruvate enters into the mitochondria, where the second and the third steps takes place. So the mitochondria is the site of cellular respiration. The Krebs cycle takes place in the mitochondrial matrix, where pyruvate is converted into various intermediate compounds. So pyruvate is converted into acetyl coenzyme A by oxidation, decarboxylation, and addition of coenzyme A. So acetyl coenzyme A is formed and it combines with a compound called oxaloacetic acid present in the matrix of mitochondria, which is regenerated. Now this oxaloacetic acid is a 4-carbon compound, acetyl coenzyme A is a 2-carbon compound. So these two combine to form a molecule of a 6-carbon compound called as citric acid, where coenzyme A is released. And this citric acid is converted into alpha ketoglutaric acid by oxidation and decarboxylation. So citric acid is oxidized, NAD is converted into NADH2, and carbon dioxide is released to form alpha ketoglutaric acid, a 5-carbon compound. Alpha ketoglutaric acid is again oxidized and decarboxylation. So this 5-carbon compound alpha ketoglutaric acid undergoes oxidation, NAD is converted into NADH2 and removal of CO2. Then the compound formed is succinic acid, and at the same time a molecule of GTP is also formed here. There is a release of certain amount of energy where succinic acid is formed. Succinic acid undergoes oxidation, where FAD is converted into FADH2. So this succinic acid is oxidized to form malic acid. Malic acid is also oxidized to regenerate oxaloacetic acid, where NAD is converted into NADH2. That is how Krebs cycle takes place in the matrix, and the NADH2, the FADH2 formed during Krebs cycle and the glycolysis, those molecules will enter into the crystal membranes, where the Racker's particles or the elementary particles are Racker's particles or the elementary particles are present, and there the third step, oxidative phosphorylation or the electron transport system takes place, which consists of a sequential series of electron carriers. Now this consists of cytochrome B, cytochrome C, cytochrome A, cytochrome A3. So NADH2 undergoes oxidation to form 3 ATP. Each NADH2 molecule undergoes oxidation to form 3 ATP. Similarly, each FADH2 undergoes oxidation to form 2 ATP. Thus, totally we get 36 ATP after the oxidation of glucose. And finally, the electrons combine with the half oxygen to form water molecule. That is the terminal oxidation step. So that is how the aerobic respiration takes place. The anaerobic respiration takes place without the use of oxygen. So only glycolysis will take place. After glycolysis, there is no Krebs cycle, no oxidative phosphorylation. The pyruvate which is formed, it will not enter into the mitochondria because there is no oxygen. So in anaerobic respiration, anaerobes we call it as organisms which perform anaerobic respiration, they are anaerobes. Aerobic respiration, aerobes. So we are all aerobes. We are using oxygen. So here, as there is no oxygen, the pyruvic acid which is formed in the cytoplasm, it may convert into alcohol or lactic acid. So thus there are two types of fermentation. When alcohol is produced, it is alcohol fermentation, where pyruvate is converted into alcohol by reduction and decarboxylation. If the pyruvate undergoes only reduction, it will produce lactic acid. There is no decarboxylation. So this is how the anaerobic respiration takes place, that is the fermentation takes place. Then there is amphibolic pathway and respiratory quotient. Cellular respiration is considered as amphibolic pathway because it involves catabolic reactions and anabolic reactions. The anabolic reactions are seen in the Krebs cycle. There are some steps of the Krebs cycle where they appear to be anabolic, so they are anabolic reactions, and the other part of the respiration shows it is catabolic. So both anabolic and catabolic are seen in the respiration, hence it is called as amphibolic pathway. After studying the types of respiration like aerobic and anaerobic respiration, so there is one last aspect which is called as respiratory quotient. So respiration is an amphibolic pathway. So respiratory quotient is also called as RQ value. Now RQ value is defined as the volume of CO2 evolved to the volume of oxygen consumed. So in a respiratory reaction, how much amount of CO2 is released and how much amount of oxygen is consumed. So that ratio is called as RQ value. So for any carbohydrate you take, the RQ value is always 1. So that means when you study the equation of the respiratory reaction, so this 6 CO2 plus 6 H2O plus energy. So this is the equation of aerobic respiration. So the volume of oxygen, the volume of carbon dioxide is taken into consideration. So the ratio of the volume of the carbon dioxide released to the volume of the oxygen consumed is called as respiratory quotient or RQ value. For any carbohydrate, it is always 1. That means the volume of these two are equal. But for proteins and fats, it is always less than 1. Less than 1. But for organic acids, it is more than 1. It is more than 1. So that means when we say that for proteins it is less than 1, it means the oxygen consumed will be more. When there is more oxygen consumed than CO2, automatically the value will be 0.8, 0.9, 0.7 and like that. That is for proteins and fats. But for organic acids, it is always more than 1, like 1.1, 1.2, 3, 1.4, like that. That means here the carbon dioxide released will be more than the oxygen consumed in that reaction. So this is what is RQ value. So the RQ value is defined as the ratio of the volume of CO2 evolved to the volume of the oxygen consumed in a respiratory reaction. So this is usually applicable for the aerobic respiration, not for anaerobic, because in anaerobic oxygen is not used, isn't it? So that is why it is applicable to aerobic respiration. For any carbohydrate, whether it is glucose or any other carbohydrate, the volume of CO2 and oxygen will be equal. So the amount of oxygen which is consumed will be equal to the amount of the carbon dioxide released in that reaction. So for proteins it is less than 1, that means oxygen consumption is more, and for organic acids it is less than 1, that means it is more than 1 for organic acids. That means the when it is more than 1, it means that the carbon dioxide will be more than that of the oxygen released. So that is about the respiration in plants, which involves aerobic and anaerobic respiration.